This is Daniel Klimmeck. I'm editor at the EMBO Journal covering stem cells, cancer, and other topics. And it's my pleasure to have an interview here with the organizer and co-organizer of the upcoming Keystone on cellular plasticity. And this is Ophir Klein from UCSF. Ophir, so I had the pleasure to join the last meeting as well, which was 1.5 years ago. That was the first of its kind, but probably many participants of this year's meeting this is their first time joining in for that Keystone. So what's the overall idea with the new format? It's rather recently introduced.
Thanks, Daniel. Yeah, so I want to acknowledge first of all my two co-organizers for this meeting, Ramesh Shivdasani from Boston and Stacy Heppert from Cincinnati, with whom we've been putting together the program, and we've really actually built a lot, as you alluded to, on the previous work done by the first group of organizers, who were Jason Mills and Mike Sander and Ben Stanger. And they really initiated the idea of having this meeting, which I think is very exciting because the concept of cellular plasticity, which is really the ability of a cell to acquire either a new identity or adopt an alternative fate, depending on how you define it, is becoming something which is emerging as a very important concept in both disease and various aspects of damage repair and homeostasis. And while the phenomenon has been addressed in part in many different contexts, in cancer and tissue homeostasis and other kinds of meetings, this was, as far as we know, the last one was the first meeting of its kind to address this question.
And that was also my impression, right? That it's really, I think the fascinating part, that it's really scientists with quite diverse background coming together and discussing this this relevant point. But you know, from an editor's perspective, the plasticity comes in many different flavors, right? So you have the flexible molecular interactions. It's at the cellular level, and then you have the tissue level you mentioned. So will there be a fair mix of those topics at the current meetings as well?
The field has advanced somewhat since the last meeting. There's a lot more, as you'll hear at the meeting, a lot more molecular information about plasticity. I think we've seen a lot of progress over the past few years in going from observing the phenomena to understanding them across a number of tissues. One of my interests is in the intestine, and we've seen in that field a lot of advances over the past few years. And I'm also glad that you mentioned the editor's perspective, because one of the things that we're really excited about at this meeting is that we have really nice representation from editors of a number of great journals, and we're planning, as you know, and as the audience will find out, to involve them in a very exciting way in this meeting.
And of course, we're happy about that. But you mentioned the advances. So I mean, they will always come with new methodologies and new technical feasible aspects. So I remember from the last meeting, that was beautiful. There was we learned about fusion organoids to address you know, interconnectivity of different organs. There was a talk on optogenetics to explore signaling. So because you have the preview, right? So you've been skimming through the abstracts and the talks. So what's the emerging, can you tell us a little about what's the emerging themes coming up in the field?
Yeah, there's a few. I would say one that we are highlighting is changes in chromatin. That's clearly a very important part of changing cell identity and cell fate is modifying the chromatin. So there's several talks that are looking at this question. Another area that we're highlighting is inflammation. Mm-hmm. And that's important in both wound healing as well as in various disease conditions and cancer. So that's a theme that we'll see some of the speakers discussing. Mm-hmm. And technically, you'll see even more single cell biology than you did at the last one, because that field mm-hmm. has advanced significantly over the past few years. And clearly, that's a really important way for us to identify the fate of an individual cell, is to understand its transcriptome.
Mm. What's your view on the imaging side of the things? I mean, the tissue, for instance, you're a tissue researcher, that there's exciting new microscopy and other means to I guess the live imaging is probably something which should be considered too.
Thank you for asking that, because that's actually something that I'm going to talk about in my talk a little bit, and some of the other speakers will be bringing live imaging. It's as you point out, it's really important to be able to follow the fate of a cell when you want to understand if it's plastic, and one great way to do that is by watching what it does.
Well, I'm sure that's going to be fascinating. How about the technicalities? I think, I've been now joining in for one virtual meeting several weeks ago, but many might have not. So I think there's many question marks popping up, right? So how will it work? Can you tell us a little more about, the sessions and the talks? Will there be question and answers live after the talks?
Yeah, so we're in a very unusual time, and the need for the scientific community to be able to communicate remains, even though it's difficult for us to travel now. So what we're trying to do is make this as interactive and open as possible. There is, I don't know if you would call it a silver lining, but one element of the meeting is that because it's virtual, it means that more people from more places, especially those who might not always have the resources to be able to travel, can participate. So that's something that we're hoping to highlight a little bit. And definitely there will be plenty of opportunities for interaction, both at the virtual posters as well as at question and answer sessions. And one addition that we're making to this meeting is having even more. There were some panel discussions at the previous one, but here we're trying to bring that to another level and actually have them at every session. And that will hopefully be moderated by editors as well as some junior scientists. So that's something that we're bringing new to this meeting.
And the poster sessions, how will they work? I mean, this is kind of a quite unusual format, right? Usually you enjoy going from one to the other, you keep chatting, maybe you have a beer even along. So how will that work now? Is there?
The way that this will work is that each poster presenter will be able to post a short video as if they were presenting live in person, and then there will be the opportunity for participants to ask questions, and then other folks can view what the questions are in the chat function and what the responses are. So that will make it quite interactive.
Yeah, that sounds good. But and how about work which is not, some of the most interesting work, it might not be presented at the poster actually, right? So is there, of course, from journal perspective, it's interesting to think about where would be the place to, where the scientists can pitch their recent story, or usually that's the bar. Will there be a virtual bar to talk about science?
Yeah, so while obviously this is not an in-person meeting, so we don't have a virtual bar, but we do have the opportunity for participants, both speakers and others, to meet with journal editors, and the keystone organizers are putting together the way that will function.
It should be fruitful, yeah. And yeah, for the younger participants of the meeting, let's say, late PhD students, early postdocs, yeah, it's unusual times, as you said, right? So what would be, do you have advice for them on how to make most out of this this meeting? Yeah,
well, like we were saying before, one potential upside is that for those students who might not otherwise be able to attend, either because this is perhaps a little bit outside their field or because there are financial considerations, this format will enable more trainees to participate. Also, of course, the costs are much lower. And in certain cases, what we found with some of our departmental seminars in-house is that some students actually feel more comfortable using the chat function and asking a question in a live room. So hopefully students and postdocs will take advantage of this capacity and put questions into the chat, and maybe that will actually increase some trainee participation. And in addition, we're also organizing a career roundtable that the trainees can participate in, and hopefully that will be of interest to them.
So that is for the academic context, or this is also, I guess this is then PIs or maybe editors as well? Or?
So we'll be organizing a career roundtable that will include both perspectives from academia and industry, and hopefully publishing if the editors participate. And also we'll have both more senior established folks and more junior people who are relatively new to their position.
I was really interested in that point you made about that the virtual format might be even, you know, it has it's kind of, we would all like to meet physically, obviously, right? But it does have its advantages, right? And it might be even more integrative in its sense, right? So I was just, you know, I'm in Europe and we are flying over to the US to the meetings, and I was talking to someone today in the UK, and she was telling me that basically, you know, you can only afford to go to so many Keystone and Gordon meetings per year, right? And maybe she's going to 1 or 2, but then with the virtual format, you save all the travel, you save the hotel, and that might enable you to actually, you know, participate in quite a few more. And then I think that's actually beautiful. And I was wondering whether also it could be that there's a shift in diversification of the background and countries of participants attending the meeting. That should be beautiful, actually.
Yeah, those are all really good points. There are pros and cons to doing this virtually, and you've mentioned many of the advantages, and having a more diverse audience, having people who wouldn't otherwise be able to attend, having people from outside the field who might not necessarily be willing to give up a week of their lives to do this, but would go ahead and jump on a virtual meeting. Also, many of us, if we go to meetings at places that are far from our houses, we often spend a significant amount of time quite jet lagged, and that affects participation. So all of those are advantages of the virtual format. There's also challenges, which the main one being that we're social animals and we like to be around other people. And I imagine that after we come out of this period that we're in, that there will probably be some kind of hybrid formats that emerge, because there are some elements of this that are very positive and that people will want to keep. And so I imagine that our colleagues at Keystone are thinking quite deeply now about what things will look like in the future. But I would not be surprised at all if future meetings have some virtual component to enable those who have small children or for other reasons can't necessarily travel to participate in the scientific community.
Ophir, one more question. This is about the interdisciplinarity of the meeting, which is key, I think, right, to spark new ideas. And so the last one, as I noticed, was a back-to-back with the development and disease keystone, right? And I thought it was really beautiful. What is your ideas about which fields would be important to attach and to show interface to from the tissue plasticity angle?
Yeah, so we've thought a lot about how to make this meeting as interdisciplinary as possible, for exactly the reason that you're mentioning, which is that the field of tissue plasticity necessarily draws on development and disease and cuts across organs and species. And the way that we've thought about this is trying to, in any given session, combine either different types of organs, different types of biological processes. We have one session which is across species, trying to compare invertebrates and vertebrates. So that's a theme that we've really tried to highlight in this meeting, is that to understand plasticity, we want to draw on the broad variety of activities that are happening in the scientific community, looking at all these different animals and organs to try to see what are the commonalities and, interestingly, what are the differences.
That is really nice. I assume, you know, then also the panel for that session will be quite diverse, right?
Yeah, and hopefully people on the panel will be able to draw from each other's experiences and get new ideas for their own research.
Yeah, beautiful. All right, Ophir, thanks so much for taking the time. It's close to the meeting, so you must be really busy. And well, we're all looking forward to learning more and getting the latest on tissue plasticity.
Yeah, and thanks for taking the time to do this. I'm looking forward to virtually seeing you there.
All right, see you there. Cheers.